High quality biospecimens really do lay the groundwork for everything we do in personalized medicine. They provide accuracy and reproducibility in biomarker identification, and they ensure that we truly understand both the molecular and cellular makeup of diseases. These samples can also give us a chance to better validate biomarkers as they're used in clinical validation assays. And they ensure that at the end of the day, our diagnostics and therapies are much more precise. But I have to say, I think the magic really comes into play when we look at how biospecimens are paired with comprehensive clinical data. So, for example, when we track a patient's outcome along with their molecular profiles, it drives much more effective personalized medicines. We also shouldn't ever underestimate the importance of a controlled clinical collection. So, when you think about it, cutting down on those pre analytical variables that can skew results is really important. So simply put, quality samples gives us very dependable insights. It ensures and enables drug discovery, clinical assay validations, and provides more actionable multi-omics insights and analyses. So at the end of the day, it's really a win-win for both research and for patient care. You need to make sure when you're thinking just about that initial collection step that you're doing that very swiftly and accurately. It's really important to think about how that initial step can have a tremendous influence on biomarkers. Things such as freeze thaw cycles can really impact how easily you detect biomarkers. And of course, there's some known implications, and I probably hesitate to say probably some unknown implications for not having tight controls over that. Another thing to think about is storage conditions. So, of course, there's not a one size fits all approach for biospecimens. You really need to think about what's the specimen type, the matrices that I'm working with, and make sure that your storage conditions and protocols are tailored to that specimen of interest. And also thinking about the research applications of the clients using those specimens and what they may be looking at and what's important to capture and, again, build into that storage condition protocol. Another thing to consider is making sure that you have really rigorous quality checks in place. So looking at simple things like size and color, doing further downstream analyses to look at viability or genetic material integrity, all of that has to be considered and everything really should get scrutinized. So we actually use a Perio digital pathology slide scanners that create a full slide image of all of our tissue sections. We pass that on to our board certified pathologists, and they actually verify that every sample is meeting our histology quality benchmarks. And those can include basic things like just confirming the diagnosis, to more quality related aspects that could be looking at the processing and making sure that the sample looks as we expected, or what's the percent tumor within that sample. So ensuring that we keep these strict audit schedules and also creating that feedback loop. So as we're looking at the specimens within our labs and doing those QC checks, we're feeding back to the sites. How do the samples look? Is there something that you can improve on? That's really what helps us turn something that can be variable into reliable. Consistency is absolutely key. It's really what makes the anchor for reliable biomarker research. So first off, when we think about consistency, it's important to speak to standardized operating procedures or SOPs. And you want to make sure that those are in place really for all parts of the process, whether we're talking about the collection aspects, the sample processing, or the storage conditions. You name it, it all has to be in there, it all has to be documented. And speaking of documentation, you want to make sure you're pairing that with really detailed documentation and that you're capturing all of those minimal technical data elements that are involved in the process. So that could be anything from the timestamps for how a specimen is transitioning from one stage to the next, logging the transport or storage conditions and temperatures. It's really critical that you're capturing all of those elements. And then you want to make sure that you have a really robust data management system in place so that as you're collecting those data points, you have a place to put them, you have the consistency, the standardization, the accuracy to ensure that you're reducing any errors associated with that sample collection and the data points. Secondly, I think it's important to call out training. Obviously, that's a really critical step as you have these procedures. You want to make sure that your staff is properly trained. And when you think about your workflow, that you're thinking about staff that are involved in all aspects. So whether that's at the collection site or within the lab, making sure the staff is trained and that you're checking in on them to ensure consistency in those procedures. That's really critical. And then finally, I think it's important to call out having a really robust quality control system. So when you think about looking at a specimen, how do you validate that all the processes were followed, and making sure that you have QC measures in place to check in on that so that that sample is really high quality and is performing in a way that you expect. So when you combine all of these measures, you're really setting that groundwork for um, biomarker studies that are much more accurate, much more reproducible, and that really overcomes a significant pain point that we find researchers cannot be all, very often encounter in their research. And it can be especially difficult as they transition from an assay development to a clinical application. What sets BioIVT apart? I think a good place to start is when we look at our certifications. So we operate under both ISO 9001 and we also have CAP certifications. And those are actually gold standards for the biobanking industry. But here we actually like to go a bit deeper than that. And so for us, every biospecimen that we're collecting actually goes through multiple layers of quality checks. And that can be everything from the histology review to the pathology confirmations. And one kind of important statistic that I like to call out, and it really keeps our clients coming back to us because they know the asteran tissue is famous for its reliability, is that what we've observed over our 20 years of collecting biospecimens is that we have a reported less than 1% issue rate with our specimens. And that's incredible. So that type of reliability is really something that, like I said, researchers seek out and find value in. I think something else that stands out is really the emphasis on traceability and having traceability in our specimen records, as well as our compliance documentation. So those blind and consent forms, and making sure that clients have transparency to that. For us, we really want to make sure that we're always being customer first. And so sometimes we have examples of clients coming to us maybe a year or years after procuring a specimen and needing to get that documentation. They have maybe questions back from a regulatory agency, or they're asking different research questions and need to take a closer look and don't have the documentation at their fingertips. So we always keep that. We have very careful documentation management systems. And I think it's really one of the benefits that BioIVT provides to researchers, that we can quickly and easily bring that up and make sure that science doesn't stop, and that we're able to have that rapid turnaround and really traceability with the documentation that we provide.